concept here. And this important concept is a passive process, and that's called diffusion. Okay? So 2 things about diffusion. 1, it's passive, all right, which means it does not cost any energy. All right? It is ATP free. That's my favorite. And then 2, it involves a concentration gradient. We're going to go from an area of high concentration to an area of low concentration. All right, when we're dealing with substances, solutes, whatever, what have you. Okay, so high to low. Good so far, right? So that explains, all right, when we're going to be dealing with oxygen, hormones, and nutrients, 3 things that flow in your blood in pretty decent amounts, all right, and their end goal is to get to the tissues. Okay, so we want oxygen, nutrients, and hormones to diffuse from your bloodstream into the tissues. Where at the same time, all right, remember the circulation is a transport vehicle. We want to pick up, it's like a garbage man. Well, I shouldn't say garbage man, but it's going to serve a role as a garbage man. It's got to pick up the trash from the cells. So it's got to get carbon dioxide, which is the waste product of respiratory gases, all right, and wastes, urea, things of that nature, okay? So what's going to pick that stuff up? So it's going to pick up stuff from the tissues, all right, and move it from the tissue into the blood, okay? So there's a couple things that affect, all right, this process of diffusion, all right? Remember what we said, all right, way back in chapter 4, actually chapter 2, all right, in 210, all right? Some things can move directly across the plasma membrane, and they don't have an issue. There was 2 characteristics. You folks remember. I didn't hear you. Small. I heard small. And nonpolar. That's right. Small and nonpolar. They're so small they can fit in. All right. It's like a mouse going underneath the door. Sneaks right in. Okay. So small and nonpolar solutes can move directly through, and the only thing that prevents them from doing so, all right, is the concentration gradient. Okay, so as long as there's a concentration gradient, they're going to move down that concentration gradient. So definitely small solutes. The example here is oxygen. All right, that's awesome. So it's going to go right through the endothelial cells, or in between them. Okay, the larger solutes are going to have some problems, unless we're dealing. Remember we talked about capillaries. There's 3 types of capillaries. All right, there's the continuous, the fenestrated. And that's the capillary that had the holes in it. Okay, these tiny holes all throughout, little holes here. Okay, and then you have the sinusoid. Well, basically, the sinusoid, instead of having tiny holes, you have like these big, long gashes. Okay, so those big holes, those big, long gashes, it's much easier for the larger solutes to pass through those. Okay, so as long as there's holes, all right, the larger solutes aren't going to have an issue unless they're too big for that hole, okay? So it's the fenestrated capillaries and the sinusoidal capillaries that have those larger spaces or gaps. All right, proteins are big, by the way, okay? They are big molecules, which is good because we saw that it's the proteins that help keep the inside of a cell negative when we're dealing with the electronegativity of the cell. They're too big to escape, all right? So that's why it was a big deal if potassium was coming and going. All right. All right. So let's talk about another way to move, all right, through cells or by cells, and that's vesicular transport. Well, guess what? Vesicular transport, that's an active process. That does require some energy, okay? But there's 2 components, pinocytosis and exocytosis. Pinocytosis, remember we called that cell drinking? All right, phagocytosis was cell eating. Okay, that's the Pac-Man cell. Well, pinocytosis is cell drinking, smaller things, more liquids are involved in pinocytosis. And then exocytosis is like a letter in the mail or a package in the mail. Okay, there's going to be something that the cell packages up, and it's going to send it to the plasma membrane. It's going to merge with the plasma membrane, and it's going to spit the contents out. Okay, so that's what we're going to see here. So we saw vesicular transport in a pretty decent role in the thyroid gland. The follicular cells undergo vesicular transport. That's how it brings iodine from the bloodstream into the follicular cell. And then the follicular cell is going to package it up and spit it out into the follicular lumen there. Remember the colloid that sat at the center of those follicles in the thyroid gland, okay, where the thyroglobin was supposed to be. Okay, that's similar to what vesicular transport is going to be. Okay, so there's a substance outside the cell. It drinks it up, all right, transports it across the cell, and then it gets secreted on the other side by exocytosis. All right, so this is going to go in both directions. So it's either going to go from the blood into the tissues or from the tissues into the blood. Okay, back and forth, back and forth. All right, this is great. You know, we learned about hormones being made, and I said, yeah, this gland will make the hormone and put it into the blood, but we never knew exactly how. This is how. Right? These hormones, for a perfect example, will be utilized through this fashion, and fatty acids, and we'll talk more about their role later on when we get into metabolism, but it's going to be the same type of scenario when we get into the liver, okay? Because the liver is going to help package up some of these fatty acids, add some cholesterol to it, and then we're going to make some awesome lipids, HDLs, LDLs, all that fun stuff. We'll talk more about that later. Well, now you're going to know, because if you ever get your lab results and you look at it, you're like, oh, your good cholesterol is high and your bad cholesterol is low. We're going to talk about that, like what makes this a good cholesterol, what makes that the bad cholesterol. Yeah, it'll be interesting. So you'll know. You'll walk out of here like, dang. Eat cholesterol, especially. That's why I let my kids eat raw butter if they want, because, okay, I don't do that. But my niece, when she was a toddler, she used to take a hot dog bun and put a stick of butter in it, and she would eat it. Yes.
My mom used to catch me eating butter too.
Yeah, I mean,
She said she would have to like lock the fridge.
listen, I'm going to come out here and say it right now. You know what? When you cook, if you're into baking, just use butter. Don't use that other stuff. Unless you can't have it for, you know, trans fats are awful. So the days of like margarine and all that other stuff, Crisco, remember Crisco? It's good though. I shouldn't be saying that. That's like straight up lard. But still. All right. But butter, I forgot the famous, not Betty Crocker. Who was the other lady? No, I can't remember the lady's name. She was a big, not Sarah Lee. Anyways, she cooks it. Thank you. Paula Deen. She is a butter freak. She puts butter in everything. Now that's bad. All right. I just love butter. All right. So we already talked about diffusion and vesicular transport. That's a review. This is new. Okay. Bulk flow is new. And we're going to spend probably the remainder of the class today talking about bulk flow. Okay. So I'm going to kind of take it easy. Diffusion involves concentration gradient. Right? Bulk flow involves pressure gradients. You need to be able to identify that. That is going to drive this whole process. All right? So that means fluids are going to move down their concentration gradient. Down. Okay? Similar to diffusion, we're going down a gradient of some sort, but this is a pressure gradient, not a concentration gradient. Okay? So this is going to involve fluids, obviously. Well, what are we talking? Oh, blood. That's a fluid. All right, I'll work with that. Okay. And what's in the blood? A bunch of stuff, dissolved substances. Okay. So we're going to discuss all that and see what it has to do with. So when we're talking about, all right, these fluids moving down their pressure gradient, the direction of the movement of those fluids is going to be, all right, how much pressure. And is that pressure opposed by anything? Right? So that's what we're going to get into, hydrostatic pressure versus colloid pressure. Okay? So wherever the pressure is greatest, it's going to win. It's like a tug of war. Whatever side is stronger is going to move that rope in that direction. Right? But in this case, right, when we're dealing with bulk flow, we're dealing with two kinds of pressure. One pressure likes to push. The other type of pressure likes to pull, right? The hydrostatic pressure is the pushing pressure. That's the bully, right? It likes to push. The colloid pressure, that's the pull, okay? So you want to know that, okay? So think of hydrostatic is pushing, colloid is pulling. All right, so these two terms here we need to understand. Filtration, right? Basically is when we move something out of the blood. Since we're talking about bulk flow, we're dealing with fluid. Okay? So filtration is moving the fluid out of the blood. All right? And that's usually going to occur through the openings in our capillaries. All right? Unless, all right, you have some pretty big openings there. But for the most part, all right, for the most part, the large solutes, substances, i.e. proteins, should be blocked. They'll stay. Okay, that's important. Okay, we should not see the movement of proteins, all right, unless there's a pathology going on. Okay, so when we were talking about our circulatory system, all right, here is, I'm going to draw like an H. All right, this is the artery side. This is the vein side or venule side, and then this little crossbar guy is the capillary. Okay? All right. So when we talk about filtration, as blood is moving into the capillary, we're going to see, all right, in this area, blood pushing, not, well, you should be leaking blood, but the fluids in the bloodstream will move out of the capillary.
You don't have
I do, I'll get there. I just want you guys to marvel at my awesome artistic skills.
You look like you're struggling.
Yeah, you don't need it. I had this image in my head, but it's not coming out on the screen. I'm like, it looks way better in my head. It looks really good. All right, way better than what the book has. All right, reabsorption, I'm just kidding. Reabsorption, all right, it's the opposite. All right, so the fluid is going to move into the blood, And so that's going to occur down here. So stuff is going to move in. Okay, if you understand that, you're going to be good with bulk flow, because I'm going to describe what sets that up. Okay, so at the beginning of the capillary, on the arterial blood flow end, stuff will move out of the blood vessel. Okay, but when we get towards the vein, all right, stuff is going to move in, which makes good. Think about it. All right, we got oxygen, we got to drop it off. All right, so I'm dropping off oxygen here. Okay, but at the same time, now I got to pick up, all right, carbon dioxide. All right, so we're picking that up, the wastes. Okay. All right, so let's discuss, all right, those pressures that I was telling you about. Remember, hydrostatic pressure is the push. Okay. So this is like in your pipes in your house. All right, if you've ever watered the garden with the hose and you have the nozzle turned off, if you turn up that pressure on the hose as high as you can, that hose will fill up, it'll become stiff, all right, and that water is going to push on the walls of the hose, all right? That's what hydrostatic pressure is, all right? It is going to be, when we talk about the pressure of the blood, all right, or the HPB, the B stands for blood, all right? This is the amount of pressure from the fluid, your blood, all right, on the vessel wall. And so it's pushing, trying to think of a good way to draw this picture. It's pushing out. Out. Right? So that's the blood hydrostatic pressure. So therefore, if it's pushing out, it's going to promote filtration. Okay? Conversely, you have what's called interstitial fluid. All right? When you think of interstitial, think of tissue. All right? This is the amount of pressure that's going to be pushing in on the wall. It's virtually zero. So we really don't consider it. Okay? Tissues just don't have that much pressure. Okay, so this is from the interstitial fluid pushing on the outside of the blood vessel wall. Okay? Pretty much zero. Good so far. All right, we'll be wrapping up here in a few moments. Oh, that's from the other, that was just them talking to me, saying they missed me. All right. Let me skip. All right, so bulk flow here. The other part, remember, we're dealing with hydrostatic. That's the first part. The second part is going to be our colloid. Now, look, I'm going to interpret this for you. Colloid means, I have written up there, proteins. There it is. Osmotic means what? Osmosis. Water. Right. Okay. So in this case, we're going to use it with fluid, but mainly water, because 93%, all right, of your blood plasma, all right, is water. Okay? So that's what we're saying here. All right, so let me interpret that for you. Colloid osmotic pressure, all right, is going to be the force or the pull that proteins have on water.
Why can't they synthetically make plasma?
Why can't they synthetically make it?
Like, if you put water.
Well, well, you know what's in the plasma. You got all those proteins there. There's a whole bunch. Hormones, which is a very small percentage. It'd be tough. That's coming soon. I feel it. They're going to be able. They're going to be growing. I have a feeling about before I die, I'll be able to grow new liver. Not that my liver's bad, all right, but I have a feeling before I die, I'm going to be able to do that if I wanted to. All right. So when we break down the colloid osmotic pressure, there's going to be two parts to that. Again, we're going to deal with what's inside the vessel, what's outside the vessel. So the blood colloid, all right, that is going to be the amount of pull that the proteins in the blood vessel, remember when we were talking about it, that's what I was saying about the albumins. All right, that's why if you get liver damage and your liver is compromised and it can't produce the blood proteins, all right, that's why you get edema. All right, because that blood colloid osmotic pressure is less, so it pulls less fluid in from the tissues and it leaves it out in the tissues. That's why you get swelling. Okay? So this is how, all right, the blood colloid osmotic pressure has to do with the amount of pull that the blood has, all right, from the tissues. That comes from our blood proteins. All right? So therefore, it's going to promote reabsorption, pulling fluid into the blood vessel. Okay? Then the other part of it is the interstitial fluid colloid pressure. And again, this has to do with, all right, the amount of pull that the interstitial proteins have on trying to pull fluid from outside, all right, well, to pull fluid outside of the blood vessel, okay? So we don't have a lot of proteins there. So again, we're dealing with a relatively low number here. Okay, so for the blood colloid, that's going to be pulling fluid into the vessel. All right, the interstitial fluid colloid is going to be pulling stuff into the tissues. Remember, it's a pull. Pull, 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 pull. Can I say that one more time? All right. All right, last part here, and then we're done. No, you know what? Let's stop there. Let's stop there. And then, because this will be a great segue into next class. All right.